All right, so for this part now, we are going to assume that there are multiple genes that can affect the same character. So just as a reminder, in the previous section, we were talking about multiple alleles, but they were all within the same gene. So as an example of this, we're going to talk about skin coloration in corn snakes as an example of two genes controlling a single trait. So, a single character. So there are four possible traits. There is camouflaged, there is black, there is orange, and there is albino. So, we saw a similar situation with the rabbits, but in that case, there was a single gene that was controlling the fur color. So in this case, there's actually two independent genes. And for the purpose of this lecture, you can think about them being on different chromosomes. So there's an OB gene and a B gene. And then there is a plus allele, or there is a non-plus allele, are kind of the two possibilities. So geneticists have found that the following genotypes lead to camouflage snakes. And this dash, what this dash signifies right here is that it doesn't matter what the other allele is. So it can either be O plus, or O plus O. Both of those situations are and then the same thing is true of the other gene. So there's actually four different genotypes that lead to the camouflaged phenotype. So you can either be this combined with this combined with this combined with this, or this combined with this. So because there's so many genotypes, it's actually useful to consider use this dash in order to signify that. And what this is showing you is really just a very simple dominance recessive relationship between this gene, this allele and this allele, and this allele and this allele. So if you have either of the dominant alleles, so if you have at least one dominant allele of both genes, then you are camouflaged. If you're homozygous recessive for the O gene, but you have one or two of the dominant alleles of the B gene, then you're black. If you are one dominant allele or two dominant alleles of the O gene and you're homozygous recessive, then you're orange. And then only if you have recessive of both are you this albino trait. So let's see how this plays out. And so if you cross these two individuals together, you would produce an F1. So again, this individual, so this is very similar to the dihybrid trait in the Mendelian genetics. But in this case, you're just controlling a single character instead of two characters. So if you cross these two pure breeding individuals together, you have this F1 right here. This F1 would be camouflaged. Camouflaged. And then if you intercross this F1 to produce F2s, you can go through the Punnett square, but you would have a 4x4 Punnett square because this individual right here can produce four different types of gametes. And then the intercross would also have the same. So I'm not going to go through this right now in the lecture, but this would be the exact same situation as the dihybrid trait. And so when you group these together, you'd basically be grouping them based upon this relationship right here. As a result, you would end up seeing 9 camouflaged, 3 black, 3 orange, and 1 albino. So whenever, again, you see this ratio of 16, you should think of a 4x4 punnets, which means one of the parents produced 4 types of gametes, and then the other parent produced 4 types of gametes. So this is a situation where multiple genes control a single trait, single character. Now we are going to, this is kind of a setup then to also understand this idea of epistasis. So again, epistasis occurs when 2 genes affect a trait. So again, dominance recessiveness still applies, but now one of the gene masks the phenotypic effects of another gene. So a very classic example of epistasis is Labradors. And so Labrador retrievers come in 3 primary colors. They are either black labs, chocolate labs, or yellow labs. And so scientists have studied the genetic basis of this, and there are 2 genes that control this character, the color of their fur. One is the B gene, and then the second is the E gene. There are 2 alleles of each, and they follow classical dominance recessive relationships, and so we are going to name them capital and lowercase based upon that. So the scientists discovered that the black labs had a single dominant, 1 or 2 dominant alleles of the B gene and 1 or 2 dominant alleles of the E gene. So again, we designate this that it doesn't matter what the second allele is. The chocolate labs had little b, so they're homozygous for the recessive allele of the B, and then as long as they had this big E, then they're chocolate. And then finally, any lab that had this homozygous recessive for the little e was a yellow lab no matter what. And so again, if we did an intercross, so if we crossed a black lab with a yellow lab, so if we crossed, this would be a black lab with a yellow lab, we could produce these F1s that are heterozygote. Again, they would be black labs because they would fall within this right here. And then again, we can do a Punnett square. So these individuals could produce 4, this individuals could produce 4. And so the ratio that you get is this 9:3:4 ratio. And so this is what's meant by epistasis. So normally you see a 9:3:1 ratio. So for example, that's what we saw in the albino snakes. And so the reason that you don't see it is that there is not this class right here. So for the albino snakes, there is this class right here. So this class and this class are both subsumed into this designation right here with it doesn't matter what the B gene is. And so yellow labs combine these 2 genotype classes together. And so this is how you recognize epistasis, is because you combine 2 of these separate genotype classes together into the same phenotypes. And this is what's meant by the masking effect. This is why it's described as a masking effect, because you expect to see 4 different traits, but you only see 3 or perhaps even less. And so if you don't see the proper number of traits, then you should recognize epistasis is going on. And so then you just group the ones that are together in order to form this 9:3:4 ratio. Another example is squash fruit color. And so this is the 4 classes again. But you'll notice that 2 of these classes produce the same color, white. And so again, this is an example of epistasis because we only have 4 traits we only have 3 traits instead of the expected 4. And this is 9 to 3 to 3 to 1. And so the ratio becomes 12 to 3 to 1 in that F2 generation. So recessive epistasis, recessive alleles at one locus masked an allele at the second locus. So this is the example of the Labradors. So the Labradors are an example of recessive epistasis because this little e masks the expression of the B allele. So it no longer matters with the B allele. And that can be recognized by a 9:3:4 ratio. Dominant epistasis is when the dominant allele at one locus masks an allele at a second locus. And so that is because this masks the expression of the G gene. And that results in a 12 to 3 to 1 ratio. So the summer squash are an example of dominant epistasis. And there's actually, kind of more possibilities. So this is an example of recessive epistasis for mouse coat color. So you'll notice that this and this have the same phenotype, and so you have a 9:3:4 ratio. The squash is an example of dominant epistasis, where we've grouped these two classes together. In peas, there is an example of flower color, where you group these 3 classes together, and then that results in a 9:7 ratio, and so on and so forth. So epistasis can mask multiple traits so that you only have 2 traits, and kind of the combination is everything is possible. So you can also have like a 15:1 ratio. So conceptually, you should realize that there's a lot of different ways that you can have epistasis, and some people will go through, there's actual names for all of these different types of epistasis. I want you to know what recessive epistasis is and dominant epistasis is, and then you should be aware and you should know that there's multiple ways that you can have epistasis occur that result in these skewed ratios.